Welcome to Study Loop.
This question reviews certain sequences that can be found in mRNA to help in the identification, preservation, and the translation and transcription process. So here we could say we have our mRNA, and we can look at our different answer choices and define what each of these sort of sequences refers to. So we have Kozak's sequence. We also have the poly A sequence, or a poly A tail. We have the Shine-Dalgarno sequence. And lastly, we have the chatterbox. So Kozak's sequence is a sequence in the mRNA structure that's going to be found before the AUG starting sequence of the mRNA strand. This is going to allow for our ribosomal RNAs to help recognize where it needs to latch onto and start in order to transcribe this mRNA strand. This Kozak sequence is, however, only going to be found in eukaryotes. Moving on, we have the poly A tail. This is a numerous amount of adenines that are going to be added on to the end of the mRNA strand, and this is going to help preserve it and prolong its life inside the cytoplasm of the cell. It's going to protect it from enzymes from degrading the mRNA before it has created a decent amount of proteins from the structure. So here, Kozak sequence is for recognition, and the poly A tail or the poly A sequence is going to be for protection. Shine-Dalgarno sequence is very similar to the Kozak sequence. Its sequence nucleotides are slightly different, but again, it's going to be found right before the AUG, and it's going to help allow the ribosomal RNAs to recognize where it needs to start the translation process. However, the Shine-Dalgarno sequence is only going to be in prokaryotes. Lastly, that leaves us with the TATA box. This is a TATA sequence that's going to be found in the DNA of the cell. So here we could say we have our TATA, and this is to help our RNA polymerases recognize where it needs to start the transcription process. So this is more a feature of the DNA rather than the mRNA strand, but it's for recognition as well. So for this question, we're looking for the recognition on an mRNA strand in eukaryotes. That's going to help the ribosomal RNA bind. So our best answer choice among these is going to be the Kozak sequence. The poly A tail is for protection rather than recognition. The Shine-Dalgarno sequence typically only appears in prokaryotic cells, and the TATA box is for DNA recognition for the RNA polymerases. So A is going to be the best answer choice here.